sí, probando sonido 1, 2, 3, sí, sí, sí. Bueno, sí, sí, 1, 2, 3, probando sonido 1, 2, 3, ¿ya? ¿Sí, ya?
El doctor Sarukhán y el mío propio les queremos dar la bienvenida a esta sesión de los viernes de la evolución que, como ustedes saben, normalmente tienen lugar en el Colegio Nacional, pero como la intención del Colegio Nacional es promover hasta donde sea posible la divulgación de distintas áreas de la cultura, desde luego que incluyendo la ciencia, dijimos, bueno, hagamos una sesión en la Facultad de Ciencias que está muy cercana a la Facultad de Veterinaria y de esta manera tal vez podamos promover la interacción entre dos comunidades que desafortunadamente rara vez interaccionamos. Y el tema de la charla de hoy es el complemento natural de una que tuvimos el año pasado de la evolución de la evolución y la genética de los perros. En esta ocasión, la conferencista tenemos la buena suerte que sea una especialista con un reconocimiento mundial de la genética y en particular de los gatos y en particular la genética de la domesticación de los gatos. Como ustedes saben, en los últimos años ha habido un interés creciente en el problema de la domesticación de plantas, en la domesticación de animales, y los gatos constituyen un modelo animal espléndido porque tienen una serie de padecimientos que son equivalentes a los que presentamos primates como los humanos. Esto los hace especialmente fascinantes porque se puede trabajar con ellos, se pueden comparar los resultados, tienen una historia atrás que históricamente podemos recoger muy bien. Sabemos que había gatos domesticados en el Egipto antiguo, vemos las momias de los gatos, no muchos conocemos la historia de los gatos en el continente americano y en realidad lo que tenemos ahí es una riqueza de información que, como ven, requiere de enfoques multidisciplinarios. La doctora Lyons es la persona ideal para tocar el tema. Ella es de formación bióloga, trabajó durante mucho tiempo en el Instituto Nacional del Cáncer, donde se percató de la importancia de los felinos como un modelo biológico para estudiar enfermedades y es ahora profesora distinguida en la Universidad de Missouri. Ella está extraordinariamente interesada, entre otras cosas, en la distribución de genes en distintas poblaciones de gatos en el continente, en el mundo, y debo decir que es en función del interés de la genética y no en función del interés de los gatos, que personalmente detesto, con mucho gusto ayudé a participar en esta charla. Me han preguntado en otras ocasiones por qué me caen mal los gatos y la respuesta es muy sencilla, porque son como los políticos, siempre caen parados y esto es algo que estamos observando cotidianamente. So, Leslie.
Thank you. Thank you very much. Buenas tardes. I only learned a little Spanish when I was in 4th grade, so I can count 1, 2, 3, 4, 5, 6, 7, that's all. But I am most pleased to come to Mexico City. This is my first trip, and even though I lived in California, yes, I've been to Tijuana, but I don't think that counts. And it's very nice to have this opportunity. Today I will give a lecture that is not my usual lecture. Usually I speak about inherited diseases in cats, but I've had to do some studies on the domestication of the cat and the population dynamics of cats so that I could apply that to our studies on genetic diseases. You need to know if your populations are the same ethnic group or of the same race from the point of view of only genetics, so that we can do our studies properly. So I have studied cats from all over the world to help us with our genetic studies in inherited diseases. Now, I'm from Missouri, but originally I grew up in Pittsburgh. I am the home of the Pittsburgh Steelers, yeah? And so I am a big football, but also a very big baseball fan. And then for 14 years I lived in California, but now I come to you from the University of Missouri. It's a very wonderful place to live. Now, historically, I have helped to identify many mutations that cause cats to be sick, but also their coat colors and their traits. I helped with the genetics of the first clone cat. This is many years now, so CC, maybe she's a bit old now, maybe she has passed away, but it was the first time a companion animal was cloned. And then also I've been involved with transgenics. So we can do transgenics cats, and we could possibly make knockout cats. If you've heard about CRISPR, it'd be very possible for cats because we understand their reproduction very well. We understand reproduction of cats better than we do dogs. So it's very possible to do CRISPR knockout cats. But mainly, I study cats that are inbred. And these are usually because the more close the population, and the population could be a random population, but just so there's lots of inbreeding, we tend to see the frequency of rare recessive diseases more frequently. So it's not because of the breed that you have rare recessive diseases. Anyone can have a disease, but because of the population dynamics, we see them more frequently. So I generally study fancy breed cats, And cats are, again, are different from dogs in many ways, that only for maybe 150 years have people really bred cats for what they look like. And mainly it's for colors and for shape and size, not for function. So dogs generally have had function and a big selection on their genome, so their entire genome has had very strong selection to form behavioral traits, where cats, not so much. So that's why we say cats are maybe not so domesticated, because they have always done the behavior that we want, and the behavior that we want, up until recently, has been to kill mice, to kill vermin. And so they have done that for 10,000 years, and so that's why we say they're not so domesticated. Now, many of our cats, what we have to realize is most of the cats of the world are not fancy breeds. In the United States, which has a very high amount of fancy breed cats, maybe only 10%, so that's still very low compared to dogs. Most dogs are of some breed in the United States. And then if they're a fancy breed, the Persian cat is the most common and most similar cat. So out of fancy breed cats, over 70% around the world are the Persian cat. So this is a very popular breed and one we have studied many times for different things in our research. But what about the regular cat, what we would call the barn cat or the feral cat, the street cat, moggy? These are everywhere in the United States, and so over 8 million cats in the U.S. and probably just as many here in Mexico. But in the United States, about 1 in 3 homes have a cat, and generally if you have a cat, you tend to have 2.2 cats. Right? So this also means that people will have cats, but they will feed the cats that are outside as well. Right? And so a vast number of cats are what will be the feral cats and the random bred cats, and less than 10% are cat breeds. So what about their evolution? We have all these cats, but really, where did they come from? Who are they? Who are these little animals that we love? And so let's start way at the very beginning, and that is the age of mammals. So the age of mammals started to occur about 65 million years ago. So in many places, different mammals started to develop. And so over the time, we're really going, this is the present time, we're going to now really focus on this aspect as far as the domestication of the cat. But at 65 million years ago, what we have to say we're studying mammals, but also the placental mammals. And the placental mammals really developed in Laurasia, and so that's where the precursors of the domestic cat are really going to start their evolution. And at about 65 million years ago, all the supercontinents had broken apart. And so each continent is developing their own mammals, including Madagascar. That's why the animals are so unique on Madagascar as well. So here we can see about 60 million years ago, all the continents have separated. Now, in that time, now they're also now going to come back and bump into one another, and then we'll have animals transfer across the continents as well. But most all our cats developed in Eurasia. So 65 million years ago, we have the development of mammals. We start to see our first cat-like individuals at this time. And you can see that even though cats and dogs are all considered to be carnivores, they are very different. So over here is the cat-like carnivores, and over here are the dog-like carnivores. And so what makes a cat in evolution? We'll see that this one lineage here, which was very cat-like, actually died out. So if we think, what makes a cat? How is it unique? How would you define a cat? Well, they have a shorter nose than dogs would, so their skull will be slightly different. They're carnassials, so the teeth in the back, they are very highly specialized to be a complete carnivore. Cats only eat meat. They need meat for their energy. So where humans use carbohydrates for their energy, cats use protein for their energy. Where dogs can eat both vegetables and carbohydrates, cats are obligate carnivores. They must eat meat. Most of your carnivores walk on the flat of their foot, except for dogs. But most others, such as the bears, they walk on the flat of their foot. So cats are digigrade. They walk on their toes, right? And then also you can see they have retractable claws. So generally when you see the footprints in the dirt with a dog, you can tell that it's a dog because you'll be able to see the claw, where with a cat, the claw is retracted and you'll never see claw marks in footprints for cats. But one of the main things, these are 3 different skulls. The Saber-Tooth Tiger, many people recognize Saber-Tooth Tigers. This is the current day Siberian tiger. So just about the same size. These cats went extinct about 10,000 years ago. This becomes an important time for cat evolution. But this is the biggest cat ever of all time. And so it was the American lion. And they went extinct also about 10,000 years ago. And probably not because of the Ice Age, but probably because peoples came over from Asia and started to devastate a lot of the animals that were living in North America. So, so this shows you that the biggest living cat right now is the Siberian tiger, and it's about the same size as a saber-tooth tiger. So again, the dentition is very different for cats. So always they will have this number of teeth, the different incisors, the canine. So here's the incisors, the canine, and the premolars and the molars. And they're very, very highly specialized so that they rip and tear meat. And we can see the difference of the American lion versus this is a regular kitty cat. So quite different, but the teeth are exactly the same and the head structure is exactly the same, just smaller in size. So you would always know this is a cat. But here's the main difference. This is the main thing that defines a cat, and it's the auditory bulla right here. So it's this little chamber that allows the cat to hear very well, helps to collect the sound that comes in from the ear into here, and this is the eardrum right here. So this bulla is actually in 2 parts. So it has 2 chambers, and the chamber is divided by a bone, and that's what makes a cat unique. So, so when other lineages of cats have died out, they've been called cat-like, and that's because this bulla was not the same. And so when you find a skull and you open this and it's 2 chambers and it's a bone, that is what is the defining characteristic for a cat. So we can see that some lineages actually died out, and really about 21,000 years ago, this is where we have the development of what will become the modern day cats. And you can see the Smilodon, that is the saber-tooth tiger. Many of these lineages died out, all these here, but now the modern lineages, which and some will lead to Felis, and Felis is the small cat lineage, and that is where we have our domestic cats. So present day, we have about 36 different species of cats. In South America, or there's, I think, a new species has been defined for some of these little tigrinas, and so a different species has been defined now. But mainly, we want to talk about our domestic cats, which are in the Felis lineage. And so you can see cats have been around for about 10 million years, really starting to develop about 21 million years ago. But the current lineage is about 10 million years. And this rather recent and rapid radiation to fill all the different niches throughout the world, every one of the niches seem to have a top carnivore, and that top carnivore tends to be a cat. So the scientific classification, just all the way from kingdom down to our species, which we'll talk about, Felis silvestris catus. So the small cat, the little domestic cat, where did this cat come from? Well, what we have to keep in mind, okay, what else is going on with the rest of the world at the same time? And what we need to keep in mind is the last glacial maximum was about 26,000 years ago. And at that time, many places in Europe were covered with the ice sheet, but many of the high places, such as the Alps and some mountains in Italy and of the Pyrenees as well, were also covered with ice. And so this allowed demarcation of the wildcats. So there was a little wildcat that was all around all these areas, and the last glaciation helped to separate those cats into particularly different subspecies. So that's the maximum, and so from that point forward, the ice is starting to recede. So about 10,000 years ago, we find the earliest settlements that are in the Near East in Jericho. We start to see evidence of proto-religions from artifacts. The Epipaleolithic era is about the same as what's called the Mesolithic era that's in Europe, but this is in the Levant. This is where present-day Israel is. And we move into the Neolithic, the New Stone Age. And at that time, at 10,000 years ago, this is when humans now change themselves. They stop being hunter-gatherers as much, and peoples in these regions now start to settle down and they become farmers. And once they become farmers, that begins the domestication process. So this is all very, very recent in evolutionary time, less than 10,000 years ago, 10 to 12,000 years ago. So at the time, there was a wildcat species that was all through Europe and also Asia. So there was no small wildcat in North and South America. Other felids came and populated North and South America, but the wildcat, which is the progenitor of the domestic cat, was not present. So all our domestic cats are old world. They've come from the old world and they've moved all around the world with human beings. So they got to Mexico with the Spanish settlers or invaders, however you want to look at that. So with like Brazil, they came with the Portuguese, and we can still see those genetic signatures to see where the cats originally came from. So there's many different subspecies of a wildcat. There can be as many as 17 that are actually listed, but that's because everybody that found a new wildcat subspecies wanted to name it after themselves. So we don't have actually very good genetic data on all these wildcat subspecies to really say who is a subspecies and who is not. But these are the common ones that people tend to accept. The European wildcat being Sylvestris sylvestris, the African wildcat, which means northern Africa, so not sub-Saharan, northern Africa, is Felis libica, and the Asian wildcat is known as Ornata, and also there's a Chinese cat called Bieti as well. So we kind of think these are different subspecies of cats, and we have done studies to try to figure out which ones are the progenitor of our domestic cats. So historically, this is where all these wildcat species live to keep them separate and alive. So again, the historical range of where all the wildcat species were. But of course, as human beings have expanded and developed into larger civilizations, these areas become more fragmented, and hence the wildcat species have a harder time moving around different areas and are somewhat unique. So the sub-Saharan African wildcat is a bit different, so that's Felis caffra. Now, what we need to keep in mind is with the domestication of the cat, it occurred where humans developed civilizations, where we started farming and settling and staying in one place. And that happened at least 3 different places, a 4th different time in the Mexico area in Central America. So we put that aside because, yes, that was a different agricultural development, and there was no wildcats here. So the domestication of the cat could not happen with the agricultural development that was here. But so in the Fertile Crescent everyone tends to think, okay, that's where farming has started, and certainly is one of the earliest sites. But also the Indus Valley, and so that is present-day Pakistan, so right on the border of Pakistan and India, and also the Yellow River region of China. That is known as an independent area of domestication as well for rice and other plant species. So I've always wondered, could there be 3 different potential domestication sites for cats as well? Because we have seen that for dogs and most all of our other species as well. Cattle have at least 2 domestications. Indicus is very different from Taurus. Horses have many different sites they were domesticated. Chickens, many different places throughout the world as well. So is the cat unique in that they have one major domestication site? And all this involved, so we have to keep remembering that our science is all interlaced and interdisciplinary. So I talk a lot with a good friend of mine is a wheat geneticist. And so it really came to play that because of the domestication of wheat and millet, these plants were available at the right time in the right place after the Ice Age, and humans started to settle down. They discover that they can cultivate wheat, and then hence they have a constant food supply and don't have to move around as much. So if there's a constant food supply, that means you have grain stores. And so if there's going to be stores of food, who's going to take advantage of the stores of food? Well, the mice and the rats. So the mice and the rats are the first to discover, hey, if we go near humans and tolerate humans, we can get lots of free food. So as animals, what is our job? Our job is to procure food for the least amount of energy, the most efficiently that we can, and make little babies. That is our job as a species, right? So the mice are thinking the same thing. Why be out in the forest looking hard for food when I can come and get a lot of food if I'm near humans? So the grain stores, but also we have to remember our refuge piles, our garbage, also attracts mice and rodents as well. So rodents come, and then the cat thinks the same thing. So now you have wildcats in the area. They're in the forests and they're in the desert regions, and they realize that there's more and more rodents. So the first cats must think, if I am bold and I can tolerate being around a human and get close to them, then I can get easy food, and I will be able to have lots of babies, and I will progenerate better than my fellows out in the woods and the forest. And so pretty much it's a symbiotic relationship between the cats and humans, that if I come to tolerate humans, and they tolerate me because they see I'm doing a good thing for them as well, then we begin the process of taming and domestication. So taming perhaps coming first, and then on to domestication, where we argue, have the cats really gone all the way to domestication? So what is our archaeological evidence of domestication of cats? There are some. Most of it, of course, comes from Cyprus. So we do have just a single molar that was found at Jericho and a whole jaw that was found at Cyprus. But the one that is really the most strong is about 9,000 years before present by the French scientist Vignier, found a whole skeleton. And so this skeleton seems to have been buried with a young child, so implying that cat was important to the child and to the family, and so this is perhaps the first evidence of true taming on the road to domestication. Now, there was recently also a site discovered in China as well. I was very excited about that. I was like, yes, this should be true. This is also around the Yellow River, but this ended up, once they did the analysis of the bones more extensively, this was a leopard cat. So that's a different species, Prion oralis bengalensis. And so that was maybe an example of taming, but we've never domesticated the Asian leopard cat. So now where are we? We have cats all over the world, and I've been able to go to many different places to find cats. And of course, we had the large grain stores in Egypt, certainly known for loving cats and actually worshiping some aspects of cats. But if you go to Egypt, all over the streets of Cairo, there are cats. And so, unfortunately, on the one hand, cats are devastating to the natural flora and fauna. But on the other hand, they are now currently important to the ecosystem because they keep the rats and the mice under check. And if those explode, then we have problems like bubonic plague and things like that. So originally, they're not supposed to be in the environment, but now they play a very important role in many ecosystems around the world. So the Egyptians certainly worshipped cats, and that's because they were very loyal. Stories about cats are written by the first historian, Herodotus, saying that a cat would run into a burning structure to retrieve its kittens. So people really admired how well a mother was and how dedicated they were to their kittens. And also they were the goddess of fertility as well. So apparently cats were being very successful at that time. You go to Rome, there's cats all over Rome, so many different places, all through here. If you look very closely, there's cats all over these ruins. They used to be infested in the Colosseum, and I think they actively made an effort to remove the cats from there. So here we can see some of them just laying around the ruins in Rome. So we can go most anywhere and find feral cats that have been there for thousands and thousands of years. Even at the Taj Mahal, I have found that Muslims are quite fond of cats, actually. There's a saying that Muhammad so revered the cat that he would not disturb the sleeping cat on his cloak. So many Muslims revere cats, not so much dogs. And their coat colors are actually slightly different in India as well. Now, also, besides archaeological work, we can trace cat domestication and their popularity through artwork as well. And so many of these are from Renaissance and also Chinese presentations from the 12th and 13th century. So we know the cats are around and they're part of people's everyday lives, but look, they're always with a rodent. They're doing their job, and that is to kill the rodents and keep the vermin under control. In Thailand, the different coat colors of the cats, these were actually, they called them different names based on their different coat colors. And here's one of the earliest depictions of a cat in artwork. And so this is an Egyptian artwork, and we can see this is actually from about 1900 and up, 2000 BC, so 4,000 years ago, this cat is displayed in artwork in Egypt. Now in Egypt, once the Egyptian culture was actually starting to decline, and the Greco-Romans were taking over Egypt, say around 500, 600, 700 BC, that is when all the mummification of cats occurred. So the priests were losing power, and they developed these votive offerings. And so you could buy a very cheap mummy, or you could buy a very expensive mummy of different animals for worship. So thousands and thousands of cats were actually mummified. Many tens of thousands of cats were mummified for these votive offerings. But we do have cats from Madagascar, and we discovered that these cats, even though the French until recently controlled Madagascar, the cats, the original populations that get there are the true cats, and they stay there. So the original population was actually cats from the Arabian Sea area. So we genetically can tell these cats apart. But notice I have some island populations, cats even from the Galapagos. But look, I have a big missing spot here, so it'd be very nice if you have a cat. We need to get a DNA sample from your cat. Okay. So my lab has worked on some studies where we're looking at DNA variation, generally known as single tandem repeats, short tandem repeats, STRs, also known as microsatellites. So we've used microsatellites, and we've also now moved into using SNPs, single nucleotide polymorphisms, and also you'll see we've done mitochondrial work. And so really why I'm trying to discover what is going on with cats is because I want to know how are populations different. When I do my studies for genetic diseases, I need to set up cases and controls. So I need to know that my populations match. And then also when we develop genetic resources, such as a DNA array, we want to make sure the DNA array is useful for all populations of cats, not just one breed or just the cats of North America. So I have taken the effort to discover how different are cats. And truly, we can see that there are very distinct genetic populations of cats. And we tried to include those all when we designed our DNA array and when we've done our sequencing studies as well. Okay. So this is just an example of microsatellites, several different microsatellites on one gel at a time. We don't use slab gels so much anymore. We use capillaries now. But here you can see a more inbred population where there's not as much variation, where a nice random bred population of cats will have a lot of variation. When we just compare the genetics of cats and we do an algorithm and just say not a priori, we don't say where the cats are from, we just say put the cats into different genetic groups. What do we find? So with our first studies, we found that cats from Western Europe, and so you see Brazil, well, these were Western European cats first, and then they went to Brazil, are different from the cats of the Middle East, and are also different of cats from Kenya. But actually, these Kenyan samples were collected off two small islands that are off the east coast of Kenya, so actually in the ocean. And these end up being a different population that we call the Arabian Sea population. So these cats would be very similar to the Madagascar cats as well. And then the most distinct is Southeast Asian cats. They're very, very different, and we see that many of our breeds come from there as well, such as the Siamese and the Burman. And so we can also look at the amount of genetic variation, and so we can see that some breeds have very low genetic variation. That's the white bar, and the dark bar is inbreeding coefficient. And so some of the inbreeding coefficients are very high in our breeds, but in random bred populations, the genetics is very strong, very diverse. Cat breeds are very diverse. And we can see that most of these are from Israel and Egypt. And so this was some of our first evidence that if you try to find the most genetically diverse populations, and if we really look at which populations have the most private alleles, we will find those in the Middle East. And so that was our first implication that cat domestication probably occurred in the Middle East and in the Near East and Israel and Turkey and perhaps Egypt as well. Now we've gone on since those preliminary studies, and we think there's about 10 to 12 different genetic races of cats. And we can see that cats from Iberia, Portugal and Spain, and the island of Majorca are genetically distinct. And so then we actually see that those cats contribute to Brazil. So we know that's a unique population, and that's different from the rest of Western Europe. Northern Europe is very distinct as well, such as the Nordic cats of Northern Europe, as well as different areas of Southeast Asia and Egypt. Iran and Iraq are very unique cats as well. So it would be very interesting to look at Mexico. So is Mexico going to be Spanish cats, so Iberian cats, or have they been infiltrated by Western Europeans? And so will they be Western European cats? So that'd be a very nice project for a student here, and I'd be very happy to host a student or have you do most of the work and provide you with our data sets for the comparisons as well. So one of the first studies that focused primarily on domestication was done by Carlos Driscoll, and Carlos was a colleague of mine. I used to be at the National Cancer Institute, so once I got my PhD, I went to work at the National Cancer Institute, and they worked on cats as well. And that's where I got to know Carlos and Dr. Steve O'Brien as well. And so they did a study where they looked at both STRs and mitochondrial DNA as well. And you can see they have several hundred cats in every category and many different living wild cats as well. And so what they found with their study is that this clade 4 was most of the African wild cats, and I think that's what it is, and that's where the domestic cats were as well. Whereas one of the other clades is actually where the European wildcat is. So you would think, oh, the European wildcat, surely that has contributed to the domestication of the cat, but truly not so much. So at the time when the cats were first being domesticated, it was more of a subspecies that was more North African wildcat-like and not Western European wildcat. So this was the first evidence that this was the species. The North African wildcat is the primary contributor to the domestication of the cat. Now there's still European wildcats. There's still perhaps wildcats in Scotland, but they have lots of introgression. So now it's very hard to study these populations because there's so much introgression with domestic cats with the wildcats as well. So they are in the forests in the areas of Europe, but we have to be very careful to be able to find the cats that are true wildcats. And so we've also done some mitochondrial studies, and so really we've been interested in forensics as well. So no cat can commit a crime without us figuring out who that cat is. But it is very interesting. I said a third of homes have a cat. And so cat fur can easily be transmitted during a crime, either onto the person or if they've wrapped if they've killed somebody and they've wrapped a body in a blanket, or there'll be cat fur, and that can help you identify who potentially did the crime. And we do have a few examples of that. So we have studied mitochondrial DNA as well, and we found very similar findings as Dr. Driscoll, and that was there's maybe 5 to 7 major mitochondrial types, but about 1 in 5 cats actually has their own unique type as well. So there is a database for, and this is the control region of mitochondrial DNA, and we can see here's the major types that are just named A, B, C, and D, and there are some derivative types as well that are just one step away from these major types as well. So we do have good databases for mitochondrial DNA for the cats. And we ourselves then use this because everybody's, okay, what about mummified cats? What's going on with mummified cats? Well, Egyptian mummified cats are very hard to study because 1, it's very hot, and then 2, all these mummies were just jammed into old burial sites and caves and tombs. And actually, one of the places I got to visit was the tomb of Maya, and Maya was the wet nurse for King Tut. And so her tomb then got just all jammed full of these mummified cats, and sometimes they spontaneously combust and cause a fire as well. So it's very hard to study cat remains because there's not very many in the permafrost. They're not preserved very well. Cats were not up there. They didn't want to be in the ice. And so you'll find lots of wolves and dog bones, but it's very hard to find cat bones. But from these bones, we did our best to isolate control region of the mitochondria of the cats. And what we did find is that the types that they were having were the types that are most common in the Middle East, and not genetic types that were common to Western Europe or the Americas. So that helped us say, okay, we don't have contamination, but then also that probably the cats of the pharaohs are the progenitors of the cats that are living today. And so those cats running around Egypt and are really descendants of the cats of the pharaohs. So now more recently, in the past 2 years, Dr. Eva Maria Greikel from France has done a wonderful study where they did get a hold of many different cat bones, and they were able to amplify portions of the mitochondrial, but it was not the same as our study. We did control region where they did the same as the Driscoll study, and that was the ND5, 6, and cytochrome B genes that are found in the mitochondrial genome. And they were able to get some sequence from these cats, and they just recapitulated the same thing, and their bones are collected over different time periods too. So it helped you to not only discover what was the species that was there, but also at what time point were they spreading out through Eurasia. And so they also looked at a coat color mutation, which causes the cats to have the classic tabby pattern, the big swirls on their side. So with these studies, as you get to more modern cats, you can see that they could define what different species that they were actually identifying. So their Sylvestris cats that they had were modern cats, but you could see they could actually identify them in some of the bones of the most ancient bones they had as well. And so overall, this study also recapitulated that probably the African wildcat was the progenitor of the domestic cat as well. And also with their study, so with their ancient DNA, they were able to show that also this has been a very complex process and a very long and slow process, where dogs had very, very strong selection on them. Cat selection has been very mild over the many eons. And so also with the classic tabby pattern, we could see in the early Egyptian art, it was more mackerel. Mackerel is the stripes or spots, where this classic tabby pattern is very unique and is a new mutation. So it's occurred during domestication, and so they were able to identify that only in some of their most recent samples. Now, historically, there have been studies where people just went out and looked at allele frequencies. So before we could do any hard genetics as far as finding mutations. So in the 60s, many people throughout the world just sat down and looked at the allele frequency of recessive traits for cats. And we could see that it looks like this blotched tabby pattern actually first developed in the UK. Because that's the highest frequency, and then it spread to different parts of the world. Where if we conversely, say, look at the orange coloration of cats, the orange coloration of cats started here and then spread to different parts of the world. So just by looking at coat colors and allele frequencies, we can understand some of the migration and domestication of the cat. Now about our breeds, what we have to remember, came from the cat populations that were in the area, right? So cat breeds come from their local regional population. And so we can see that some have fairly high genetic diversity, where some are getting lower and their inbreeding is not so well. So cat breeds will actually outcross to different breeds and to random bred cat populations, where that would never actually occur in our dog breeds. So this does help to keep our cat breeds a little healthier than many of the dog breeds. So cat breed selection really only started occurring in the 1900s. 1871 is the first cat show, which Charles Darwin actually attended, and that was at the Crystal Palace in England. And at that time, they had maybe 5 different groups of cats. They had long-hair cats, and we do know that the long-hair mutation actually came from Persia, so the cats are named Persians. The long-hair mutation actually is on the background of a short-haired haplotype that is most common from the Middle East and not common in other parts of the world. So there were those cats, and then there was foreign cats, so cats that came from Thailand, and those were the pointed cats we now know as Siamese cats. So those were in the cat show, and then British short hairs were in the cat show. And then in 1906, we had the first cat show in the United States, and we added the Maine Coon. We had to have our own cat. But really, it's not our own cat because it came from Western Europe, but it came with the Pilgrims. And so we had a cat in the United States for a few hundred years. That was named the Maine Coon. But also the Manx cat was one of the first cats. That is the cat with no tail. The Manx cat was recognized as a different breeds. So the different breeds at the time were not really breeds. They were different populations of cats that we could see were genetically different in some way. So since that time, these are the first 5 breeds that were noticed, and we've had an explosion of the cat fancy, but primarily most of these breeds have developed after the World Wars. So during World War I and World War II, most of the cat fancy just went away, and most of the breeds were lost. So really, now that we have maybe 40 to 50 breeds, most of those have developed in the past 50 to 75 years after World War II. And most of our cat breeds are just different by coat colors or fur types. Again, not selected on behavior, even though we do have some behavior differences. A Siamese cat is very, very different from a Persian cat, but we haven't primarily selected on behavior. So less of the cat genome is under selection than, say, a dog or a cow or a pig, where those are very complex traits that we select for meat quality or milk production. So if we look at our breeds, we know that the cats from Siam, the Thailand cats, now present-day Thailand, the cats from Burma, the Burmese, are truly, their closest relatives are the street cats from those regions. And so there are very genetically distinct cats. And most cat breeds have developed from Western European cats. A few, the Egyptian Mau, the Turkish Angora, and the Turkish Van, have developed from the Mediterranean areas as well. So mainly 3 different regional locations where cats can be identified. So this is different from dogs. If I take DNA from your cat and I do my genetic test, I can tell you what worldwide regional population that cat came from. You can't do that with dogs. Dogs have kind of regressed, having a lot of breeds, then became a lot of mixed breeds, and then those became the dogs that are on the street. Whereas with cats, the cat populations were there, and then we developed the breeds from those cat populations, and the cat populations still exist where they originally lived. So many different breeds are just different by one mutation, long hair versus short hair, so I don't really consider that a different breed. We'll see genetically that's true as well. There's many curly-coated cats, of all things. Why we have so many Rex-type cats. The Lycoi is a new cat that we have discovered, and Lycoi in Greek means wolf, so it's the werewolf cat. And then also there's naked cats, and this is not the same as the Mexican hairless dogs. That was the first thing people tried to look at, and it's like, no, it's different. And then just more coat colors, lots of tabby stripings, which is fairly unique to cats. We don't see that so much in dog populations. And then also we have funny physical traits such as curled ears, folded ears, and little short tails. Now, some of these I study because there are biomedical models that are associated with them, such as the Scottish fold. It's a type of osteochondrodysplasia, so it's kind of a model for arthritis in humans as well. And then we have different sizes. We do have a dwarf cat called a munchkin, which just have the short legs, as many dog breeds, but not the same gene. And the Singapora is maybe the smallest cat, and the Maine Coon is the biggest cat. A good, healthy Maine Coon cat can be 10 kilos in size. If we look at our breeds as far as genetic structure, we see that many breeds are not genetically distinct. So these are, I mentioned in the beginning, Persians were the most popular. So Persian cats are the progenitor for many different breeds, and so we can see that their genetic signature is very strong. So overall, if we look at all the different breeds of cats, there's maybe 20 to 25 genetically unique breeds of cats, and the rest are just single gene mutations and variations on the same theme. So again, I mentioned Persians are the most popular, and they have contributed to, but now becoming very popular is the Bengal cat, and that's the hybrid cat between the Asian leopard and the domestic cat, which I won't discuss today. Again, the Persian is most common worldwide. And why? Is because many people like the brachiocephalic look, the short face. So this is the short face of a Persian versus the long face, so this is a bit longer than a normal cat, of a Siamese cat. And people tend to like this better. I don't know, the Walt Disney characters have big eyes, this neoteny, the look of a baby. And so many people just use the Persian. Instead of doing long, slow selection, they cheat and go fast by using the Persian as an outcross. And so we can see many breeds have now the look of what a Persian cat has. And so also many breeds such as exotic shorthair, British shorthair, Scottish folds, Selkirk Rex, these are all have the same genetic signature of a Persian cat. Now, Cinnamon. Cinnamon is our sequence cat. He's our Abyssinian, and we now currently, at this moment in time, because we have a PacBio long-read genome assembly for the cat, at this time, the cat has the best genome assembly than any other species other than humans and mice. We don't count those. But for cattle and pigs, the cat currently is the best. So we're very proud of that at the moment. And we can see the size of the genome is about the same size as any other mammal. We generally say 3 gigabases, 3 billion base pairs is generally the average of what we say. And so what now, as we've used that genomic information to help separate out what is different about cats, we know that cats have gone on to develop many interesting aspects for hearing and for vision. They're very specialized. And actually, their hearing range is more broad than a dog, and so is their vision range as well. But also, cats have traded off some of their olfactory genes where they don't use smell as much as they use their vomeronasal organs, so pheromones. So they have expanded their repertoire of pheromones. So cats are always, what does people always get rid of cats? Because they pee everywhere, right? So they're peeing everywhere because they're putting pheromones out, and that is their job. That's what they're supposed to be doing. And so we can see genetically that they are very specialized for their pheromone receptors. And so we can see there's differences between the different felids and a lot more genes for their pheromones as compared to their olfactory. So olfactory smell has dropped a little bit, the pheromones has gained. And then everybody's always looking for the genes that are involved with behavioral traits. And I am a little bit skeptical because if you do one of these selection studies, you always tend to find these genes, but nobody really shows the proof. And so I am a little skeptical even about our own paper that yes, we found these genes involved with behavior and neurological development. Yes, that makes sense because those would be the things that would change an animal's behavior to be more bold and to be more calm around humans. But we really have, no one really has gone on to show any proof that this gene has changed in this way and that has created the domestication process. And so with that, we are getting, as our genomes get more and more, we get more sequences from more cat genomes, we are going to be able to do better selection studies and have more controlled studies where we actually look at very fine-tuned behaviors. So dogs have been sold on behaviors a lot, but we haven't found a lot about behaviors. We have found obsessive compulsive behaviors, things that are biomedical models and are specific variants, but we're still working very hard to figure out the complex traits that affect an animal's behavior and hence their domestication process. And probably it is going to be one of the hardest things to do in a cat because they truly are not as domesticated as many of our other species. Defining domestication, a cat can be released and go into its environment and be fine. It will survive. Where dogs need to pack hunt. And so if you release a dog, it might be fine, it'll be eating the trash, but it won't learn to hunt for a few generations until it reacquires its ability to form a group and to become a pack and to be able to communicate within that pack. So that's why another reason why we say, okay, dogs are more domesticated, where cats, you just turn them loose and they're fine, they're going to survive. So with that, that's the story of cat domestication. I think we all agree and know that they are only partially domesticated, and this was the story why. So thank you very much for your attention. And I'm very happy to take any questions.
Tenemos tiempo para preguntas, por favor.
I'm in no rush.
Thank you very much. That was a really nice talk. Have you studied or seen a study related to zoonosis on cats? I mean, something that you mentioned that they're not really domesticated. Still, they're around us. Is there any kind of study that relates some disease that passed to cats, from cats to humans, or do you have any kind of genetic information related to this topic?
Yeah, we're actually involved with 2 studies right now. So if we look at the public health aspect of cats and zoonosis, one thing that's always been attributed to cats is toxoplasmosis. However, if we're under argument as far as this right now, because most cats that live in a household and never go outside do not have high risk to produce toxoplasmosis. And they shed the eggs very rarely, only 1% of cats. So everyone knows if you're a pregnant woman, do not clean the litter box. But that risk is tremendously low. The risk is more from eating uncooked meat and poorly cooked meat. And yes, cats are the primary host for toxoplasmosis, but truly we can easily overcome that hygiene aspect of it. So that is one thing we've been involved with. Cats can get the flu, they can get plague, but they're not very good hosts for it, so they don't transmit it very easily. Something in the United States is called bobcat fever, cytozoonosis, and that is one that we're studying. That bobcats are fine. They get this disease from ticks, and they're just fine, where if a cat gets the disease, they will die. But we do find a few cats that survive. So I would like to look at the genetics of what is different between the survivor cats and the cats that most cats succumb to disease, and then what is genetically different with a bobcat that they can deal with this disease. Same thing with the AIDS virus. Cats have lived with the AIDS virus for a very long time, so most cats do not get sick from the AIDS virus unless they're compromised by leukemia. If they get leukemia, yes, they're going to get sick and die. But usually cats with the AIDS virus are co-infected with leukemia virus as well. So some cats can die of the AIDS virus, but it's not, like many shelters will get rid of cats because they're FIV positive, but that's not a high risk. That's not a high risk for the cats. Yeah. So there are some zoonoses. Cat scratch fever, Rocky Mountain fever. But again, that's from the dirt of the fleas, and the cats just transmit it via scratch. Yep.
Can I make a question?
Yes.
Actually, I remember about 10 years ago, there was a report of a higher amount of retro elements in cat genomes.
Yes, so many species. So if we think about our genomes, there's 3 billion base pairs. Only 1 to 2% of that is coding DNA that makes proteins. The rest is we don't know. And a lot of it now we're discovering as we genome sequence individuals, we'll find that maybe 30 to 40% of the time we can find the mutation. It's in the coding part of a gene. But a lot of the times we're finding, oh, well, there's regulatory elements everywhere, and that's in the non-coding regions. And we do find that endogenous retroviruses have inserted themselves into many places that disrupt these regulatory genes, and then hence are affecting our cats. And some are in all felids, but yes, domestic cats have quite a bit of actually mitochondrial DNA dispersed into their genome as well. So about 16 kilobases of the mitochondria is in many places of the cat genome. And then endogenous retroviruses, sign and line elements are there as well. But there's one specific to the cat called RD114. And I was just recently looking at that because I thought that was now going to cause a coat color in cat, but we eliminated it. But yes, cats have many endogenous retroviruses.
Si hubiera más preguntas o comentarios, incluso a favor de los gatos.
Thank you for your talk. Is there some investigation of the microbiome of the cat?
Some people are starting to think about that. I think primarily, I do know some investigators at UC Davis. So cats die when they get old, if they don't get hit by a car or eaten by a coyote. They get old and they die of renal failure or intestinal lymphoma. And it is often thought that maybe inflammatory bowel disease is a precursor to intestinal lymphoma. So there is interest in the microbiome of the cat to try to understand. You also have to realize that cats have only been on commercial food for the past 50 to 75 years, since the 1950s. And our commercial food, although the pet food companies do a very good job of responding to the needs of the cat, such as when they discovered taurine deficiency that caused heart disease and blindness in cats, they were very quick to adjust the food. But still, cats should not have that much grain in their foods, and so we see that cats tend to have more problem with diarrhea and inflammatory bowel disease. And so there's a lot of interest from pet food companies to try to understand the gut of the cat genome. And then also cats have a lot of inflammatory responses. And so the gut is our biggest immune organ. And so there's interest, but I haven't paid attention to know what's happened with those studies. Who has a cat? Okay. Who has a dog? Who has a cat and a dog? Okay. Who has a fancy breed cat, like a Siamese or a Persian? See, very few, yeah, very few. But good, I'm glad you all have cats. Extra cats.
I have a question.
Yeah.
Yeah, there are those very fancy, well-known experiments in which a Russian team tried to domesticate foxes, and you could see very obvious morphological changes in the foxes in very, very little time. Has anyone tried to do something equivalent with wild cats or anything of the sort?
No, we haven't. Actually, Anna, who's behind some of those studies, was just at MU, I think, last week or so. She's now at University of Illinois. And so what do we see out of those foxes? One of the first things that happens is the acquisition of white spotting. And we see that in cats as well. And it's very interesting because the white spotting is caused by a retroviral element in the intron of the gene KIT1. So it's in intron 1, and it's interesting because a small 800 base pair, just the LTRs of the retrovirus are there. The small 800 base pair insertion causes dominant white, where a big 8 kilobase insertion causes less white, the bicolor white spotting and stuff. So we do see that there's definitely selection for color traits. And whether that is associated with, the cat being more bold, I'm not so sure. But I am fairly convinced with, does anyone have a tortoiseshell cat? A cat that's black and orange? No tortoiseshell cats? These are very known. Tortoiseshell female cats are known to have a bad attitude. All right? So tortitude is what they call it. But the orange male is a very, very loving, nice cat. And so is this people's perception or is this true? And I've had the own experiment of my feral cats around the house. And so I do think that orange might be, it's on the X chromosome, and so is MAOA, which is involved with serotonin. And so I do think that maybe some of the cat colors are maybe associated with some of their domestic behavior. But no one's ever done the study to prove it. Yeah.
Bien, si no hay más preguntas, yo creo que le podemos agradecer a la doctora Lyons su excelente charla. Thank you so much, Leslie. Y los quisiera invitar a la siguiente sesión de los viernes de evolución, que va a tener lugar en el Colegio Nacional, Donceles 104, la entrada es gratuita, y va a ser el viernes 11 de mayo a las 6 de la tarde, a las 18 horas. La va a dar el doctor Víctor Moreno Mayer, que es un egresado del Centro de Ciencias Genómicas de la UNAM de Cuernavaca, y él está trabajando ahora en Dinamarca, en el Museo de Historia Natural de Copenhague, sobre la genética de la población original americana. Han estudiado el grupo de humanos que existe todavía con características genéticas SNPs, y esto muy bien definidos, en Alaska, que aparentemente se separó en dos poblaciones. Una se fue hacia lo que es la parte este de Estados Unidos, otra siguió hasta la Patagonia, y él es el primer autor de un artículo espléndido que se publicó en Science hace unos cuantos meses. Entonces, como siempre, la invitación es abierta para todos ustedes y pues muchas gracias por su presencia. Hasta luego. Thank you so much.
Thank you.
Convinced me to hear it.
Almost. They're superior. That's why people don't like them. They're superior.